For every major scientific breakthrough, there was a time before it. A time when the world was one way, and then a time when it was different. A time when the sun revolved around the earth. A time when all bacterial illness was untreatable. A time when dots and dashes wrote our hellos and goodbyes. A time when all computers were the size of suburban homes. And for all those times, before and after, historians were both witnesses and recorders. Chemists and engineers, physicists and geneticists. Scientists from every discipline mixed a desire to discover and create, with a willingness to question the status quo. Each made the decision to push forward, spurring some of history's greatest innovations. The Science History Institute is the organization dedicated to collecting and sharing the scientific achievements that have shaped our lives. We document and commemorate the history of chemistry, chemical engineering, and the life sciences, telling the stories of innovators and inventions, experiments and analysis, pioneers and their crowning achievements, researching and preserving the past, so that we may inspire a deeper understanding of the world we live in and what the future may hold.
Good afternoon. I'm David Cole, President and CEO of the Science History Institute, and welcome to the 2022 Heritage Day Awards ceremony. This is the Institute's 18th Heritage Day and the second one we are presenting virtually. And we are very excited to be welcoming viewers from across the country and indeed the globe to today's program. Heritage Day is a celebration of exceptional achievements in chemistry, chemical engineering, the life sciences, and allied fields and industries. And this afternoon, we honor the various accomplishments of three individuals: Dr. J. Keasling, Dr. Brian Hoffman, and Dr. Barry Siadat. This past year, as we have all started to emerge from our homes and get back to our offices, our lecture halls, and cultural centers, the Science History Institute has reopened our doors again. Scholars from all over the world are studying in the Othmer Library, conducting research on a vast range of topics in the history of chemistry, and the museum is open to the public with our newest exhibition, Downstream, which explores the over 200 years of water analysis and protection in the Delaware Valley. And our staff is back roaming the halls every day. We are so very excited to have our community back with us again. In spite of the hardships during the COVID-19 pandemic, the Science History Institute has made so many new connections with our digital audiences. We've been able to reach more of you than ever with our virtual lunchtime lectures, our monthly Joseph Priestley Society programs, the Distillations podcast, and our innovative online exhibitions with Google Arts and Culture. We are excited to continue to roll out engaging and accessible digital content for our constituents all over the world, even as we are coming together again in person. At the center of all this work is the Science History Institute's mission to collect, preserve, and share the remarkable science stories that reflect the diverse history of scientific inquiry, discoveries, and the everyday lives of scientists that are not often shared. And during today's program, we will capture three unique stories that can continue to be shared long after we finish here today. I hope to see you all in our museum in Philadelphia soon. And for those still at home, be sure to dive into our catalog of content that you can access from wherever you may be. The Heritage Day program and all of our activity would not be possible without the support of the many individual supporters and organizations who contribute to the Science History Institute throughout the year. Today, I'd like to thank the individuals, companies, and groups who partnered with us to make Heritage Day possible. Specifically, the Bolte Family Foundation, the American Chemical Society, and the American Institute of Chemical Engineers. And now, without further ado, on to the awards. It is my great pleasure to introduce Francis Bolte, who will present our first award, the Richard J. Bolte Senior Award for Supporting Industries.
by an introduction. In 2007, the Science History Institute and my family gave the first of the awards to my father, Richard J. Bolte, who founded BDP International in 1966 here in Philadelphia. Today, this enterprise is one of the largest logistics companies in North America, supporting the chemical, life science, and healthcare industries, as well as the humanitarian aid sector in facilitating the safe movement of products and goods across the globe. My father was a major supporter of Science History Institute, and today my brothers and I continue that tradition through the support of the Bolte Family Foundation and board service. I'd like to take a moment to recognize our eldest brother, Rich, who this year will culminate his term as chair of the Institute's board of directors after a period of 12 years of service. To honor our father's business philanthropic legacy and continue the partnership between the Institute and my family, today we give the 16th Richard J. Bolte Senior Award for supporting industries. In years past, this award has gone to such luminaries as Frederick Frank, the first investment banker to specialize in biotech, pharmaceuticals, and healthcare sectors, Steve Holland, the former chair of Brentag, and Eugene Garfield, the creator of the Science Citation Index. Today, it is my honor to present this year's Richard J. Bolte Senior Award for Supporting Industries to Barry Siadat, co-founder and managing director of SK Capital Partners. Barry Siadat has been an inventor, innovator, business leader, and an investor in specialty chemicals and material industries. He holds more than 24 patents and is the author of over 20 scientific publications, as well as several business and management articles. He has held leadership roles at W.R. Grace & Company, Allied Signal, and Avient Technologies. Since 2000, he has led a variety of private control investments in specialty chemicals, specialty materials, and pharmaceutical companies, and has been the lead investor and chair of more than a dozen privately held companies. In 2010, he co-founded SK Capital Partners with Jamshid Kinejad with the aid to support and invest in companies in the specialty materials, chemicals, pharmaceutical industries. To date, they have invested in 29 companies, which generate revenues of approximately $14 billion annually and employ over 20,000 people globally, including a team of 57 people at the headquarters in New York. By the end of 2020, SK Capital Partners had approximately $5.5 billion of assets under management, and they are still growing. Dr. Siadat has made sure that his growth of the chemical sector is being done with an eye on the future. Under the leadership as managing director, the firm implements a rigid sustainability policy and has since the firm's inception. It is equal parts due to his innovative spirit, business prowess, and philanthropic nature that today we give this award to Barry Siadat. Congratulations, Barry, on this much deserved award that recognizes your many contributions to innovations in the chemical industry.
Hello, everyone. It's certainly a great honor to be the recipient of the 2022 Bolte Award. I would like to thank the Science History Institute, its executive team, and the members of the nominating and selection committee, and humbly accept the award. Serving the chemical and molecular sciences has been a lifelong mission for me, an interest and a passion that I started with me when I was 10 or 11 years old, when I set up a small chemistry lab in our house and started to play and learn. In high school, I continued my interest in science and mathematics, and upon graduation, when I entered college, I majored in chemical engineering at the University of Wisconsin. where I also continue to do research on materials alongside my studies under Professor Stuart Cooper. Upon my graduation from college, Professor Cooper suggested that I go to graduate school into University of Massachusetts in Amherst and study polymer science and engineering and chemical engineering, which I did with Professor Robert Lenz, where I did additional work in chemistry and material science. So as far as I'm concerned, the passion for chemical and molecular science was well cultivated in me by the time I entered the business world some 44 years ago. In the industrial world, in the industrial business world, it's hard to ignore the profound impact of chemical and molecular sciences on every aspect of us, our society, our civilization, and the mankind. From healthcare to nutrition, from electronics to communication, from the comfort of our homes to the communication, to the transportation, industry, the impact of molecular sciences and chemical sciences are profound. So it's no strange coincidence that our industry is known as the industry of industries. 96% of all products manufactured around the world depend on the products of our industry. However, we see that industrial progress by all industries have also brought with it a host of unintended consequences, ranging from deforestation to create more farmland, to climate change, to contamination of natural resources. It is once again the duty and the opportunity of our industry to help solve these problems by developing sustainable products and processes that not only enhances the safety and sustainability of our industry, but also that of all industries, and consequently our civilization and the quality of our lives for generations to come. Today, a large body of work is underway in various laboratories and pilot facilities within SK Capital portfolio of companies, as well as throughout the industry, to help all industry to be more sustainable. Just to cite a few that is being worked on in our various portfolio companies, I would add lighter weight materials for highest fuel efficiency in transportation and mobility products. Materials to improve electrical power storage and batteries. Materials to improve windmills and photovoltaic cells to harvest wind and solar energy. Better catalysts and better catalytic processes to reduce waste and carbon footprint. Technologies to reduce greenhouse gas emissions. And processes to improve recycle and reuse single-use plastic packaging. Non-polluting dyes and processes to reduce wastewater pollution in industries. These are some of the works that we're doing today in our various laboratories to help create a future for our planet that's sustainable. Our list goes on, and the industry as a whole is doing a lot in continuing to develop materials and chemicals that improve speed, bandwidth, and memory storage of digital data to enable easier, faster, and cheaper and more effective use of electronic communication and e-commerce. Our industry is developing new active pharmaceuticals with limited side effects and with in providing new therapeutic benefits. Our industry is developing carbon capture technologies to capture the carbon dioxide in the air and reduce its effect as a greenhouse gas on the planet. And our industry is developing a variety of blue and green sources of energy to replace oil and coal. So our industry is stepping up to really create a better future, just as we have done in the past, by creating a better future for mankind. The list goes on. Our science, the chemical and molecular science, is the way to a sustainability renaissance. I'm personally proud to play a role and lead our investments in that direction. The direction of building a sustainable portfolio of businesses that each and every one contributes to the health and sustainability of our planet and quality of our lives. I conclude my remarks by humbly accepting the Bolte Award for support of industry in chemical and molecular sciences, and applaud all of my colleagues who are leaders in building a sustainable world for all of us.
Congratulations to Dr. Siadat on his award here today. You can learn more about his work at skcapitalpartners.com, which is also linked in the chat. Our next award is the American Institute of Chemists Gold Medal, which the Science History Institute has had the honor to co-present since 2003 as part of our Heritage Day program. We are pleased to be able to call the American Institute of Chemists one of 35 affiliate organizations with whom we collaborate. For those of you who are unfamiliar with their work, we will now hear from AIC President, Dr. David Menuda, and see what they are all about.
The American Institute of Chemists Gold Medal is our highest award and recognizes extraordinary accomplishments in chemistry and chemical engineering in the U.S. The award has been bestowed upon 74 remarkable men and women since 1926, including Nobel laureates Glenn T. Seaborg, Herbert C. Brown, Elizabeth Blackburn, and Dudley Hirschbach, to name but just a few.
To present this year's AIC Gold Medal, I'm excited to say we have a friend of the Institute and previous winner of this award, Dr. Chad Merkin. Dr. Merkin was given this honor in 2016, and in 2019 was awarded the Perkin Medal by another Institute affiliate, the Society of Chemical Industry. From his office at Northwestern University, we welcome Dr. Merkin.
Pleasure and personal honor to be able to introduce Professor Brian Hoffman, this year's AIC Gold Medalist. Brian has been a great mentor, colleague, and perhaps most importantly, a field-leading and defining scientist for over 5 decades. Brian came to Northwestern University in 1967 and joined an emerging chemistry department and a cadre of world-class inorganic chemists. With his interest in both materials and bioenergetic systems, he quickly established a name for himself and distinguished Northwestern University in the process. His contributions are far-reaching, spanning fundamental advances aimed at understanding electron transfer and catalysis, respectively, in complex biological systems, and the development of new techniques to probe such systems that are uniquely connected with his name. Examples include, first, his extraordinary work using spectroscopic methods to determine catalytic mechanisms of metalloenzymes. Here he pioneered the use of ENDOR, which is electron nuclear double resonance spectroscopy, to identify key intermediates in previously unknown or poorly understood catalytic cycles. The breadth of impact of this work cannot be overstated. It extends to all the major metalloenzymes, including our understanding of nitrogenase and how nature splits and fixes N2, as well as how the important class of radical SAM enzymes function as H-atom transfer catalysts. A second example is his pioneering work on long-range electron transfer between proteins, both at crystallographically determined long distance and in dynamically docked protein complexes. Here, his experiments on mixed metal hemoglobin hybrids elegantly characterized extremely long-range electron nuclear tunneling, over 25 angstroms, within a protein complex of known molecular architecture. But perhaps his principal contribution was to show that transfer of an electron hole across a protein-protein interface almost invariably is controlled not by the electron transfer process itself, but by the dynamics of conformational conversion among ensembles of interface configurations. Finally, Brian has been a pioneer in the synthesis and characterization of novel, complex, and functional metallomacrocycles, especially of the porphyrin and porphyrazine class. These structures are being explored for their optical properties in the context of medical imaging. Brian has had an illustrious career that continues to flourish across many domains today. He's been recognized with some of the most important honors in chemistry, including election into the National Academy of Sciences and the American Academy of Arts and Sciences, the ACS Alfred Bader Award, the ACS Cotton Medal, the Gold Medal from the International EPR Society, the Bruker Prize for ESR, the RSC Chat Award, and the Zavoysky Prize, and many more. He's published over 680 scholarly papers and mentored an enormous number of talented and accomplished graduate students and postdoctoral associates. Only one person can win the AIC Gold Medal each year. I cannot think of a better person to select than my friend and colleague, Brian Hoffman. Making him this year's recipient is both fitting and appropriate. Congratulations, Brian.
Let me begin by thanking Chad for this concise, but really quite remarkably clear and complete discussion of our research. But with that, I'd like to really go forward to thank the American Institute of Chemists Awards Committee and the President's David, namely President and CEO David Coles of the Science History Institute and President David Minuta, President of the American Institute of Chemists. And I'd like to thank them for this singular honor. It was a few months ago that I was doing what I do most days, working on a manuscript. And when I was struggling or frustrated, I'd go to my email like everyone else does. And out of the blue came the email that announced that I had won this award. I had no expectation and I had no pre-warning, so I was quite surprised. Of course, I immediately read the award letter, and I'd like to quote from you a piece of it, which says, this award is for, this gold medal is for contributions to society through application of scientific research and contribution to the biochemical profession. Well, Chad has told you about our work, but in fact, what he told you reminds me of the quote from Bismarck, which I'll paraphrase as following. Doing science is a bit like making sausage. It's what the outcome is that counts, but the process isn't always pretty. And as someone who's been deeply engaged in the process of our research, it brought me up short to listen to Chad talk about the sausage itself, because I know what went into it, the false starts and the second thoughts. And so that was really quite a lovely way to learn about how this work is perceived from the outside. But what really brought me up short was the first thing I did after reading the award letter, which of course, as anyone else would have done, was to look at the list of awardees over the last almost 100 years. And that list of previous recipients contains friends, colleagues like Chad, and most of all, and most important of all, lots of my scientific heroes, and many of those being all of the above, friends, colleagues, and heroes. It would take somebody a lot sterner than I not to have a fleeting, at least, thought, do I really belong on that list? But the good news was that I immediately replaced that thought with the recognition that that's the wrong question. Over the years, I've worked with an enormous number of talented, creative colleagues. And it is absolutely clear that the research that is associated with my laboratory was achieved through the process that was described by two of the great classical musicians of the 20th century, Lennon and McCartney. The work that our lab is noted for was achieved with a little help from my friends. And on behalf of those friends, it is my honor to accept this award.
Thank you and congratulations, Dr. Hoffman. We salute your career and ongoing work in the field of chemistry. And now let us turn to the final honor and the keystone of the Science History Institute's award program. The Othmer Gold Medal is named after Donald Othmer, a noted chemical engineer, inventor, philanthropist, professor, and one of the original benefactors of the Institute. And just like the medal's namesake, past recipients, scientists and innovators such as Dame Carol Robinson, Sangeeta Bhatia, Joshua Boger, Gordon Moore, and Robert Langer are recognized for accomplishments in the material sciences, from leaps in research, success in commercial business, and strides in chemical education. Today, another visionary joins these ranks. Dr. Jay Keasling of Berkeley and co-founder and CEO of the Joint BioEnergy Institute, commonly known as JBay. And while I could tell you a little bit about his remarkable career, we thought it would be nice to hear directly from his friends and colleagues.
The three words I would use are brilliant, one of the most intelligent people I've ever met, committed, very much dedicated to the work that he does and the impact that it can have, and he's compassionate. He approaches everything that he does with an eye towards the good that it can do, and that's true whether or not it's his science or it's his interactions with human beings.
He's a pioneer in synthetic biology towards chemical production. He has really demonstrated how we can take synthetic biology from the lab into an industrial process.
I think the thing that Jay will be most remembered for is using metabolic engineering to try and tackle some of these really large societal challenges. I think you started to see that with artemisinin, engineering a microbe to make a reliable source of this medicine that will save millions of lives.
Many years ago, we engineered yeast to produce the anti-malarial drug artemisinin. And this developed a process that's an alternative to the plant-derived process. This can deliver therapies at a low cost. Some of the most exciting things that are coming from the field in general are the things that people can really understand and grasp and see that will change their lives.
He loves what he does, not only the research and where all the research is going to take him, but also he likes training people and helping them grow.
I've been an entrepreneur and an educator for now 30 years. And I think the way they play off each other is that I get a lot of really bright students and postdocs that come to work in my lab. And because it's a mix of basic science with application, is a good testing ground for entrepreneurial activities.
From the very beginning of working in his lab to when I decided that I wanted to start Berkeley East, he was more supportive than I could have ever possibly imagined. And it was really his confidence in me and him pushing me to go out and write that first small business grant that got me there.
I was delighted to be able to join Jay's lab as a graduate student, one of his earliest graduate students. Jay was always looking to encourage us. He never showed any frustration He never showed any impatience. And so I've looked to him as the example of how he not only conducted his science, but how he managed his research group, and have tried to emulate that as I've actually had my own independent research career with a lab at an academic institution.
He really wanted us to think on our own and own our own decisions. He gave postdocs a lot of room to try new things, come up with new ideas, collaborate with new projects. And to this day, I collaborate with former postdocs of Jay's, now each of us in our own labs.
And I think into the future, you're going to continue seeing the impact of his work on engineering microbes to make things that otherwise would have to come from petroleum.
It's really been fantastic to see the industry and the area grow. This combination of delivering products that are cleaner and the processes to develop them are better will, in the future, give us a healthier planet and better human health on top of that.
I don't think that I myself or Berkeley East would be where we are today without Jay's support.
He demonstrated without exception that it was possible to be a phenomenal scientist and also be a phenomenal human being.
Jay still has, at least 20 more years in him, so there's probably a lot more things to come.
Later in the program, we're going to be treated to a brief talk and a Q&A with Dr. Keasling. But first, another titan of science and industry is with us today to provide some remarks on this year's Othmer medalist. Dr. Bill Hazeltine is an award-winning author, entrepreneur, and scientist known for his groundbreaking work during the HIV/AIDS epidemic and as co-founder of the Human Genome Sciences. He also sits on the Science History Institute's Board of Overseers. And most importantly for today, he has also been a mentor to Dr. Keasling. Welcome to the program, Dr. Hazeltine.
It's a great pleasure to recognize Jay Keasling for his lifetime achievement, which is being recognized by the Othmer Gold Medal tonight. Jay is deserving, as perhaps no one else, of this high recognition. Jay's career as a chemical engineer has been outstanding. You might even say world-changing. I first came upon Jay's work when I was helping to develop one of the first synthetic biology products, artemisinin. I was on the board of One World Health that had been asked to fund his research. We did, and I was astounded by the speed with which he was able to assemble a whole series of genes, get them to work, basically like a concertmaster in a single cell, and produce a very valuable drug needed by the whole world to fight malaria, artemisinin. But his achievement was far more than creating one single molecule using these new methods. It was actually the opening gun of what turned out to be a race and in fact a flood of new products, ranging from biofuels to pharmaceutical products to almost anything that contains a carbon molecule. If we look forward at the implication of Jay's breakthrough work, we can see it as a definitive step forward in moving from a fossil fuel dependent carbon-based world to a biologically based carbon-based world that takes carbon from the atmosphere, creates new products, and then returns that carbon to the atmosphere, something that is very badly needed. Not only that, but extends enormously the range of biomolecules that we can make that either are identical to natural products or improve upon natural products, and even molecules which nature has never made. So there is perhaps no better person I can think of, and I know of, to receive this high recognition. Not only that, but Jay is a tireless proponent of the benefits of synthetic biology for mankind, for health, and for a cleaner environment. Congratulations, Jay.
Hello. Today I'm going to talk about manufacturing with and commercializing biology. I think that many of you know that petroleum has been the source of our transportation fuels, pharmaceuticals, and other chemicals. We pump that petroleum up from underground, refine it in a refinery, and then turn it into all of these extremely useful products that we use on a daily basis. If we look at the chemistry that underlies that transformation of petroleum to all those products, it's not only well established, but it's high yielding and produces hundreds of thousands of different molecules. And roughly $2 trillion in the US economy depends on that petroleum being transformed into all of those products. We'd like to have a future where we could use renewable resources to produce our fuels, our products, and even our pharmaceuticals. In doing so, we would take carbon dioxide out of the atmosphere, we would fix it into plant biomass, and then in a biorefinery, we would transform that plant biomass into these same useful products and potentially even new products that you couldn't make from petroleum. And if you burn those fuels in your car, your truck, or a plane, they will be returning carbon dioxide to the atmosphere and therefore will not add additional carbon to the atmosphere that petroleum does. Biomanufacturing is the manufacturing or biotechnology that uses biological systems to produce these commercially important biomaterials or biomolecules that are used in medicine, in food, in beverage processing, and in industrial applications. Now, we can do that biomanufacturing in many ways. One way is we can use microbes. We can engineer them, or we can use native, natural microorganisms, grow them in bioreactors on the biomass, and transform that biomass into all of those interesting products. We could also engineer plants to produce those products. Plants produce many of the existing products we use. They can be engineered to produce those same products. We could also engineer algae, and in this way, the two can grow on carbon dioxide and sunlight directly and produce the product. We can also use a cell-free system where there's no cell involved, no plant involved. Everything is done in a test tube or in a reactor with individually purified components. This sector of the US economy is growing rapidly. I show here the growth in this sector over 2 decades, 3 decades, and it has been growing substantially to become a significant part of the US economy. Now, I'm going to be talking to you today about the work that I've done in my laboratory to engineer microbes as chemical factories that transform this biomass into all of these useful products. And many times we take the biochemical pathways from plants or from bacteria, and we place them in an industrial host that will grow in these fermentation tanks and transform the biomass into bioproducts. I want to talk about 3 examples from my laboratory of work that we've done over the last few years. The first is on the production of low-cost anti-malarial drugs. Malaria is a problem that afflicts about 3 billion people on the planet. Roughly 200 to 250 million people have the disease every year, And every year, over 500,000 people die of the disease, and 2/3 of them are children under the age of 5. There is a therapy that's a traditional Chinese medicine called artemisinin, and it is a very effective treatment, a cure for malaria. It was first discovered by the Chinese and written about in around 150 BC, and then it's used for treating malaria about 100 BC. It was largely forgotten until the Vietnam War, and this person, Tu Yu, went out and poured through the medical literature, and she and her medical corps in China developed artemisinin as a therapy. And by 2004, it was recommended by the World Health Organization as the drug of choice for treating malaria. This is how we get artemisinin, or how we got it. It was grown on plantations, purified from the plant after a growing season. You get the pure artemisinin out of the plant, which is then chemically derivatized into the various drugs that are incorporated into therapies to treat malaria. One of the challenges in treating malaria this way is the large swings in price and availability. Starting from the recommendation by the World Health Organization that it should be used to treat malaria in 2004, the price began to swing wildly on the market. This made it very difficult for people in the developing world to be able to afford the therapy, and difficult for pharmaceutical companies to deliver the appropriate doses that are needed to treat the world's malaria sufferers. Around 2004 is when my laboratory started to get involved in engineering microbes to produce artemisinin. We would engineer yeast that would produce artemisinic acid through a fermentation process. Artemisinic acid is a precursor to artemisinin and is naturally found in the plant that produces artemisinin, Artemisia annua. Then chemists would chemically convert it into the same derivatives that are incorporated into anti-malarial drugs. This is how we engineer a yeast to produce artemisinin, or to produce any other chemical. The first step is to really find the metabolic pathway to transform natural intracellular metabolites that are inside yeast into the final product. And we use something like a Google map to find all of the possible routes from the intracellular metabolite to our final product. This map comes up with a pathway that uses enzymes to transform those intracellular metabolites into the desired final product. And then we go looking for the genes that will encode those enzymes that we can then place into the yeast to transform biomass into our desired final product. We put the genes into yeast. They are expressed in yeast. When they are expressed, they produce the enzymes in the biosynthetic pathway. And if all works as planned, we can transform the intracellular metabolites into our final product. And then we grow the yeast in tank on that biomass. The yeast produces the final product, and then we extract it from the fermentation broth and purify it and incorporate it into those therapies. In the case of producing artemisinic acid, there were some enzymes in the metabolic pathway that were known, and the genes that encoded them were known. So we could go into databases, we could find the sequences for those genes. We could then send those gene sequences to DNA synthesis companies, where they would be synthesized, and they would send us back vials of the genes that we would then incorporate into yeast. Other parts of the biosynthetic pathway were not known when we went after this pathway, and so we had to go into Artemisia annua and discover those missing steps. Artemisinin is produced in small sacs on the leaves of Artemisia annua. And we reasoned that there are cells at the base of those sacs that are enriched in the enzymes that produce artemisinin, and therefore they would be highly expressing the genes that encode those enzymes. So we went into those sacs, we purified the cells at the base of those sacs that were producing artemisinin, and then we used genome sequencing to sequence the genes and then identify the potential genes, and then we place them in our yeast. When we place those genes in yeast, we place them on the yeast's natural chromosomes that exist inside the nucleus, and we place them under the control of gene expression switches that allow us to balance all the genes in that long biosynthetic pathway. We were very fortunate that we were able to find the genes, we were able to express them in yeast, and when they expressed in yeast, they produced artemisinic acid. But that artemisinic acid wasn't inside the yeast with all of the natural metabolites inside yeast. In fact, yeast found the artemisinic acid to be slightly toxic, and so it turned on pumps to pump the artemisinic acid outside the cell. And this gave us a natural purification method for producing the artemisinin and purifying it from all of the thousands of metabolites that are naturally inside yeast and that would have contaminated the artemisinin. And when we increased the production of artemisinin at Amyris, a company that I founded and that was working directly with us on this project, the artemisinic acid actually crystallized in the fermentation broth because there was so much of it. And this gave us an extra level of purification in this overall process. We then licensed the process to Sanofi, and they developed a light-based process to convert artemisinic acid into artemisinin. And in the bottom right is a picture of the manufacturing facility in Italy that they built. Since the time of these discoveries and engineering the yeast, millions of treatments have been delivered to Africa based on the process developed in my laboratory, and that was expanded and greatly enhanced by Amyris and by Sanofi. And Huvepharma has since purchased that process and has the capacity now to produce nearly 1/2 of the treatments needed annually for the world. And on the right there is a blister pack of artesunate that contains the artemisinin that was produced with our genetically modified yeast. And since the introduction of semi-synthetic artemisinin, the large swings of price and availability that we saw earlier when artemisinin was first recommended as the drug of choice for treating malaria have subsided, and the price has stayed low and the availability has stayed high. Now, we can take that yeast that we built for producing artemisinin, knock out or remove the genes for producing artemisinin and replace those with other genes to produce other like products. So this engineered yeast becomes a platform for producing many other things. For instance, you could produce flavors and fragrances which are related to artemisinin. You could produce nutraceuticals like lycopene, which makes tomatoes red and is an important nutraceutical for humans. You could produce the $2 billion a year Taxol anti-cancer drug. You could produce sweeteners like riboside, and most importantly, produce biofuels that would reduce the amount of carbon put into the atmosphere. I want to talk about some other products that we've been working on in my laboratory since then. One of these sets of products are cannabinoids. They are naturally produced by cannabis. Two of the molecules that people are probably most familiar with, THC and CBD, are shown on the right. And these have been widely used for treating pain, relieving the side effects of cancer treatment. Extraction from cannabis is certainly possible, but when you produce therapeutics, CBD is often contaminated with THC, which is unacceptable to pharmaceuticals and to regulatory agencies. And so they must produce these using chemical synthesis. And this can be extremely expensive to do. There are therapeutics that have been produced and approved by the US FDA that are based on cannabinoids. And I show some of those there. The middle one, Epidiolex, is based on CBD and has been used and is widely used for treating epilepsy in children. And it's extremely effective. Now, one way to produce these cannabinoids for therapeutics is in greenhouses, but greenhouses are expensive to build. They take a lot of electricity for the lights, and cannabis takes a great deal of water. And so this process is not very scalable. Scaling microbial production processes is very scalable. It can be done readily and has been done over many decades. So my laboratory wanted to produce cannabinoids inside yeast in a very similar process that we did with artemisinin. This would allow us to produce both the rare and unnatural cannabinoids that haven't been explored as therapeutics, to produce low-cost cannabinoids that would require less inputs of water and energy. We were able to engineer yeast to produce a number of natural cannabinoids shown there in the middle, and a whole range of unnatural cannabinoids that aren't produced in any plant, but that might be extremely effective as therapeutics. And this technology has been now scaled and is being commercialized by Dimitrex. And they intend to produce cannabinoids for personal care, for supplements, for pharmaceuticals, and for food and beverage. And this will reduce the water and electricity inputs that are required for growing cannabis and extracting these cannabinoids. Finally, I want to close with another story about foods and beverages that are environmentally friendly. Hoppy beers are quite popular. They're very popular all over the world. And in order to make a hoppy beer, you use the yeast to brew the beer, and then you add hops in the end. And that process extracts the natural flavors from hops into the beer. Hops are expensive to grow. They require inputs, high inputs of water, and the content of the active ingredients, the flavor molecules, can be highly variable in hops depending on the variety and even on the field that they're grown in and the weather conditions under which they're grown. The two main components of hops that are responsible for the flavor are relatives of artemisinin and the cannabinoids. And we reasoned that we could engineer yeast to produce the hoppy flavor of beer without actually having to add hops. So a graduate student and a postdoc in my laboratory engineered yeast with these flavor components. They carefully balanced the genes in the biosynthetic pathway so that we would have just the right flavors in the beer. And this has now been commercialized. And you can actually buy beers from many breweries on the West Coast and now across the US that have been produced using this yeast that was originally engineered in my laboratory. And since then, they're going on to produce many other flavor components that you would find in beer and in wine. And in this way, reduce the water inputs, reduce the inputs of other molecules that you need, and shorten the production time to produce these beverages. There are many other opportunities on the horizon, and there are some very exciting companies that are exploring these opportunities. One opportunity are specialty chemicals and materials. We'd like to have flame retardant materials, but many of the flame retardant materials we have now release toxic chemicals. And so they're often not used for clothing for children, for instance. We'd like to have self-repairing materials so that if they break or are damaged, they would repair themselves. Flexible electronic materials, and even new 3D printed plastics that are highly recyclable and have new properties. With biology, we can produce many of these materials and do so in an environmentally friendly way. There are huge opportunities in agriculture. Many of the crops that we grow require large inputs of nitrogen-based fertilizers and water. In fact, the nitrogen-based fertilizers are quite expensive, and they take a great deal of energy to produce, and that results in more carbon dioxide in the atmosphere. If we could engineer plants to fix their own nitrogen or to be drought tolerant, then those plants could be grown in all parts of the world where fertilizer and water are not easily accessible. And finally, another opportunity, I showed you an example of artemisinin, which is a traditional Chinese medicine. But there are many more traditional medicines in many cultures, and those certainly have active ingredients in them that are as effective in treating the diseases that those traditional medicines were developed to treat as artemisinin is in treating malaria. Wouldn't it be great if we could go into those rare plants and fungi and pull out the genes that are responsible for making those active ingredients and engineer microbes to produce them, and thereby have effective treatments for many more diseases? So with that, I'd like to thank first the organizations that I've worked with. I've been at the University of California, Berkeley for the last 30 years, and I've worked with some extremely talented graduate students and postdocs and undergraduates, and they have enriched the research and greatly influenced the research that we have done. I'd also like to thank all the companies that I've worked with over the years, not only the companies that have grown out of my laboratory, but also the companies that have licensed our technology. I'd like to thank the research institutes we've worked with. I have many collaborators in these research institutes, and they've created a very rich environment for my graduate students and postdocs and undergraduates to work. I'd also like to thank funding. The Bill and Melinda Gates Foundation generously funded the artemisinin project. The other projects were funded by the Department of Energy and the National Science Foundation, and I thank them for that funding. It is a very unique environment we have here in U.S. science, where the federal government richly supports our basic science, even when there's no application in mind. And this is what's allowed the U.S. to become this technology juggernaut that it is in biotechnology and in many other areas. I'd like to just close again by thanking the people I've worked with. I want to start with mentors in high school and college who encouraged me to do science. And I can point to a handful who really made a difference in my life and steered me down this path of research. And I want to emphasize that to educators out there in the world, because it's extremely important that we have an influence over the people who work with us, and we can make a huge difference. I'd like to thank my graduate and postdoctoral advisors who advised me on my path that got me here to this research career. They were instrumental in teaching me what I know today and how I do the things I do. I'd like to thank the graduate students, undergraduate students, and postdoctoral fellows who've worked with me at Berkeley. I feel very fortunate to be working at Berkeley, and I pinch myself every day that I'm able to be in this incredibly rich environment. And finally, I'd like to thank my family who supported me through thick and thin in the journey that I've been on, and who've sacrificed so that I can have this incredible career in science and technology development. And with that, I'd like to thank the award committee. I'm really honored to have this award. This is the medal that I received, and it's just a huge honor for me to receive this award. And then behind me is the wonderful plaque that I have, and would just like to thank everyone who's responsible for this award. And with that, I will close my presentation.
Congratulations. Congratulations, and thank you very much, Dr. Keasling, for such an engaging and enlightening talk. I'm sure everyone enjoyed that. I understand that you've agreed to answer a few questions from our audience members who are all watching from home today. So if you're game, we have a few of those questions queued up, and I'm happy to share them with you.
Great. Looking
Okay.
forward to it.
First question from an audience member. What brought you to the exploration of biomanufacturing with yeast?
Yeah, it's a great question. And it goes back many decades. I learned about Genentech being founded when I was a kid and read about it shortly after, the genetic engineering revolution, and thought, wow, that's really cool that you could engineer a microbe to produce life-saving drugs. And it really catalyzed my excitement about this field and really brought me to the field. And then it was a course on genetics in college that really kind of reignited that passion within me and got me down this path.
Very good. Next question. How do you maintain the quality and purity of a desired chemical using synthetic biology for commodity chemicals? What are the main watchouts?
Yeah, so commodity chemicals are challenging, I think, for anyone, because these are produced at a very low price, usually from petroleum, and there's a lot of old manufacturing that's available to do it. There are facilities that are completely paid off, and so competing with commodity products, commodity chemicals, and biofuels can be a real challenge. And it means that you have to get the purity just right, as this question is kind of focused on. You have to get the titers, rates, and yields to be extremely high. But the companies that have been able to do this have been able to successfully compete. And it turns out that in many places there's a green premium. So if you can produce one of these commodity chemicals using biology from a renewable resource, that in fact, in some places they'll pay just a little bit more, and that gets it over the hump. But it's a great question, and it's really focused on the right problem. And I will just add one more thing to that answer, and that is, with a microbe, you can, in many cases, produce a purer molecule than what you would get through chemistry or through extraction from a natural producer, from a plant. And so if you can get the chemistry to work, often you can get a purer molecule than what you would through other routes.
Understood. Another question from an audience member. What are some other possible applications of biomanufacturing that are being experimented with at J-Bay that you can tell us about?
We've got some great research on producing some highly recyclable plastics from sugar. And so not only would they be renewable, coming from a renewable resource, but they're easily recycled and would replace things like polyethylene, polypropylene, polyurethane, which are huge contaminants right now. We have some fantastic work on sustainable aviation fuels. And if you think about where kind of the industry is going, we see a lot more electric cars on the road. We're seeing some electric delivery trucks, and we'll see maybe some larger electric trucks. It's going to be a long time before we see airplanes that are flying on battery power, if we ever see it with commercial airlines. And so we really need fuels for commercial airliners. And this is an area where we're putting a lot of emphasis right now, because we think it's extremely important if we want to tackle climate change.
Understood. Another question. Whenever the day comes that fossil fuels are exhausted, is it practical to think that bioengineering will be able to replace all the uses that fossil fuels once served?
The fossil fuel industry is enormous, and I touched on that at the beginning of my talk. It's about $2 trillion every year out of the U.S. economy, if you count fuels and commodity chemicals, specialty chemicals. And so to think that one technology is going to be able to replace petroleum is probably not right. I think what we're going to see is in the future, we're going to see a combination of ways to replace petroleum. We'll see solar, we'll see wind power, provided we have batteries. We'll see a lot of automobiles, passenger cars that run on the batteries. So I think where the fuels go, it's sustainable aviation fuels. That's the best place because it's going to be a long time before we have battery operated planes, as I described. And then when we look at some of the specialty chemicals and new materials, that's where biology can really excel. And in fact, produce things that we can't even get from petroleum right now. Give us new materials that are self-repairing, new materials that are highly recyclable. So I think there's a bright future in the materials and also in the sustainable aviation fuels area.
Excellent. And now we move on to a more enterprise-related question. The question is, what is the biggest challenge when moving something out of the lab and into a commercial enterprise?
Yeah, that's a great question and a key question. And I think the scale-up process itself can be really challenging. And I've seen that with some of the companies that I've founded, where going from the laboratory into a larger scale facility, you tend to scale up in larger and larger scales as you go along. And that scale-up process can often fail. The microbe may not perform as well in a large-scale reactor as it does in the laboratory. Sometimes you can be surprised and it'll outperform in a large-scale reactor, but usually that's a challenge. And it's a particular challenge for small companies because doing these kinds of experiments is very expensive when you have to do them at the large scale. We have a long way to go in terms of being able to predict better how to scale up processes, but there's work being funded by the federal government, being done by many companies in this area to try to improve this scale up as you go from the laboratory into large scale production.
Very good. This next question asks you to make a tough choice. The question is, what is the most exciting thing happening in biomanufacturing today, in your opinion?
Wow.
The single most exciting thing happening.
The single most exciting thing. Well, I'll have to tell you, that's really hard to choose from. Let me just maybe deflect a little bit and say some of the areas that I think are extremely exciting. I think the things that are super exciting, the food space is becoming incredibly exciting right now. And there's a lot of synthetic biology and biomanufacturing that's going into that. And so I think that's going to continue to be a really interesting space, especially as the population of the planet grows and we need sustainable ways to produce food. I think the new material space is also extremely exciting. We have a lot of the materials we've been using traditionally and for many decades are polluting the planet, both in the process to make them as well as when we decide to discard them. And so we need to rethink this entire space of materials and think about cradle to grave for all of our materials, because some of these are going to end up in landfills, some will end up in the ocean, and we'd like them not to be constantly polluting those places. And so we really need to think about both how we produce materials and then how they live around us and are disposed of.
The next question has to do with career paths. And the question is, for those starting out in their science careers, what is the best advice you can give based on all your experiences?
Yeah, I would say don't specialize too early. Get a broad, especially as in high school, as an undergraduate, and even as a graduate student, get as broad a base as you can under you, because the world is rapidly changing and there are going to be all kinds of opportunities in front of you. And you want to be prepared for those and not necessarily pigeonholed into one area. And I think if you work to have as broad a base as possible, then you'll be able to choose the areas of science that really excite you the most and frankly are the most needed by the planet. And I think that combination of choosing something that really excites you and something that can make a difference in the world is a dynamite combination that will keep you busy for decades.
Very inspiring. Next question, back to the science and its applications. Artemisinin had a huge public health impact. Are there other medicinal or health applications of biomanufacturing in the works, especially as we are emerging from the COVID-19 pandemic?
Yeah, I mean, it's a great question. And there are so many. I mean, if you think about the COVID vaccines, these mRNA vaccines, right? I mean, this has been an amazing science feat that they were able to develop these vaccines in this very short period of time and get them out to us and produce millions and millions of doses also in record time. Think about how many more millions of lives would have been lost if these vaccines hadn't been made available to the world. This is a tremendous accomplishment. And there are more things that are coming like this. And in fact, we've got a great project in the lab right now I can't really talk about that's going to go into future vaccines. And I'm super excited about that project. But, you know, I think this people need to look at this whole area of developing these vaccines and just marvel at what a tremendous accomplishment it was. And it really demonstrates what science can do when, you know, the right government programs are put into place to encourage it and to fund it.
We'll look forward to your talk on the next vaccine product when the time is right. We have one more question, which I'll share with you now. Are there any other ways, perhaps ones you haven't mentioned, are there any other ways that this science might be able to combat some of the effects of climate change already happening today?
Well, I think there's many ways that the science can help to combat climate change. There are some great science around pulling carbon out of the atmosphere, getting plants to pull even more carbon out of the atmosphere. There have been some great projects on that, making plants more efficient consumers of sunlight and carbon dioxide. Building microbes that will take carbon out of the flue gas of coal-fired power plants and of steel mills and other places that have a lot of carbon dioxide and fix those into products. And of course, you know, I talked about making products from carbon dioxide that's been already fixed from the atmosphere. That's yet another way. And I think sustainable agriculture is going to be a huge part of this. If we could get beef cattle to produce less methane, or if we had a replacement for beef cattle, using less fertilizer, as I mentioned in my talk on crops, would also help climate change. There's many ways that we can improve agriculture and have a big impact on climate change. And I'm super excited about that because I think it's going to be a great area for science as well as for commercial development.
Very good. Well, that was our last question. And Dr. Keasling, again, congratulations. Thank you for the terrific presentation and for fielding these questions for such a vibrant discussion. And I want to say hats off to our audience members today for asking such insightful questions. What a terrific way to cap off a day. I want to end the program with a big thank you to all three awardees and their colleagues for their participation in honoring their work. And another big thank you to our sponsors, the Bolte Family Foundation, the American Chemical Society, and the American Institute of Chemical Engineers, without whose help this event would not be possible. Thank you very much. And to all of our viewers, thank you for tuning in. We hope that you share today's final program with your colleagues and like-minded friends, and encourage you to take a moment to subscribe to our YouTube channel so that you'll know when the Science History Institute adds a new video. And if you really enjoyed today's program, please consider making a gift to the Science History Institute. Instructions on how to give easily right now are on the screen. We invite you to become a part of our growing community of supporters around the world. Thank you all so much again. Have a wonderful evening.